these RNAs don't get into protein, they just function as RNA. So, okay, so, but we are focusing on proteins, and for the proteins, we need messenger RNA, and we also need transfer RNA. So the transfer RNA looks like this in the left. There is a 5' end, a 3' end. Some of the bases can interact with each other, but some others don't, and this forms these loops in the structure. And then here on the bottom, we have like the when it's just extended, okay? And it has this section called the anticodon. And the anticodon is going to be complementary to a codon that is encoded in the messenger RNA. So when the messenger RNA arrives, it has a codon that binds to this triplet of nucleotides in the transfer RNA, and if it's the right match, it will give it an amino acid. So in this example, is GAA, it's carrying phenylalanine amino acid. And this is how the information goes from messenger RNA to a different level, which is the protein. And the proteins will be made of these amino acids, like phenylalanine. And the genetic code, it has been figured out in that there is a genetic code of 3 bases, and it's degenerate because some of the different combinations of base pairs can give the same amino acid. Okay, so there is someone raising their hand. Santiago?
Yes, sorry, Miguel, how are amino acids bonded to the RNA, and so do they interact with the sugar group, or how does that work?
Oh, so it would go interacting. They don't interact directly. It will be. Oh, you mean the transfer RNA and the amino acid?
Yes. So what's like the bond with the transfer RNA molecule?
Oh, so there is this terminal ACC in most of the transfer RNAs, and this will have it undergoes some processing. So it needs to be capped. It needs a bit of processing so that it can attach through a chemical bond to the amino acid, and it's in the ribosome where that bond is cleaved as well.
Okay.
Yes. So, okay, so as we were saying, this is a degenerate code because 4 different nucleotides, sorry, 4 different combinations can code for the same amino acid. And sometimes it's only 2 of them. But the point is that we know if we have an RNA sequence or even a DNA sequence, we know what is the amino acid. But you cannot do the reverse. If you have a protein sequence, and by the way, it's usually the DNA that gets sequenced. But let's say if you had a protein sequence, you cannot go back to the DNA or the RNA because there are many options. For example, let's say you have a serine, it could be any of those in the RNA. So it's degenerate, and but we can go in one direction, and that's extremely important for any study, because if you sequence a genome, you know if it's a protein coding gene, you know what's the sequence of the protein, predicted sequence. Okay, so the ribosome is the place where all these transfer RNA and messenger RNA interact, and this is through 3 sites called the E site, the P site, and A site. Okay, we'll see in the video next what each of these sites is doing. But essentially, it's a molecular machine that will be taking the RNA and the transfer RNA and making the catalysis of each of the amino acids to each other. So we'll go through the video now.
The job of this mRNA is to carry the gene's message from the DNA out of the nucleus to a ribosome for production of the particular protein that this gene codes for. There can be several million ribosomes in a typical eukaryotic cell. These complex catalytic machines use the mRNA copy of the genetic information to assemble amino acid building blocks into the three-dimensional proteins that are essential for life. Let's see how it works. The ribosome is composed of one large and one small subunit that assemble around the messenger RNA, which then passes through the ribosome like a computer tape. The amino acid building blocks, that's the small glowing red molecules, are carried into the ribosome attached to specific transfer RNAs. That's the larger green molecules, also referred to as tRNA. The small subunit of the ribosome positions the mRNA so that it can be read in groups of 3 letters known as a codon. Each codon on the mRNA matches a corresponding anticodon on the base of a transfer RNA molecule. The larger subunit of the ribosome removes each amino acid and joins it onto the growing protein chain. As the mRNA is ratcheted through the ribosome, the mRNA sequence is translated into an amino acid sequence. There are 3 locations inside the ribosome, designated the A site, the P site, and the E site. The addition of each amino acid is a 3-step cycle. First, the tRNA enters the ribosome at the A site and is tested for a codon-anticodon match with the mRNA. Next, provided there is a correct match, the tRNA is shifted to the P site, and the amino acid it carries is added to the end of the amino acid chain. The mRNA is also ratcheted on 3 nucleotides or 1 codon. Thirdly, the spent tRNA is moved to the E site and then ejected from the ribosome to be recycled. As the protein synthesis proceeds, the finished chain emerges from the ribosome. It folds up into a precise shape determined by the exact order of amino acids. Thus, the central dogma explains how the 4-letter DNA code is, quite literally, turned into flesh and blood. The job of this mRNA is to carry the gene's DNA.
Okay. Don't know how to exit. All right. So ribosome synthesize proteins from combinations of 20 amino acids. And in this slide, you can see the 4 amino acids and how they are bound together by peptide bonds are forming these amide groups. Now they are called amino acids because they have an amino group and a carboxyl group, group, which is an organic acid. So they have amino acid, and then they have a side chain here that can be of different compositions, and that's what makes them different in the end, the side chain. So methionine will have this structure in the side chain. The aspartic acid has this structure, the leucine and tyrosine. And in the ribosomes, where when they are carried by the transfer RNA, they will get together by either the carboxyl group or the amine group, depending on which amino acid we're talking about, but they will be held together by this bond, by this catalyzed inside the ribosome. And you saw how it happens in the model in the video. So it's really complicated process, but in addition to that, there are some proteins that will require special modifications. So even once they get out of the ribosome, they need a specific folding. And in this case, this is for insulin, which is a small peptide, well, compared to other proteins. And it will get connected and cleaved by certain enzymes that eventually give this structure. So it's important to this concept of post-translation that can occur in many proteins and actually increases the diversity of different proteins. These are X-ray crystallography that can determine the structure of the folded proteins. And here we can see the different models that are used. This is a model of just the line with different amino acids. Here on the right, there is the ribbon models where these, for example, mean beta sheets, and these loops are alpha helices. And those are motifs that are common in many proteins, because once the protein is synthesized, it will acquire a different various special arrangement, and the special arrangement tends to repeat among different proteins. So the motifs can be found again and again in other proteins, but the structure is unique. And these are space filling models that most of those are the ones in the videos where you can see all the atoms in the molecule. So to get to those structures, you need to do a technique called X-ray crystallography, and it all starts with a crystal. So after there is a crystal of a protein, has to be very pure to make a crystal in the first place. But then after this purification and crystallization, you can there is a laser beam of X-rays that will give a diffraction pattern, and through this, remarkably from this pattern, it's possible through mathematical methods that include Fourier transformation and other tools of calculus. You can actually, well, a physicist will create an electron density map, and that is the basis for a protein model. So it's really a remarkable technique to analyze the structure of these big macromolecules. So the ribosome is actually the mother of proteins, and this is similar to the mother of dragons, but it's of proteins. So it's a massive molecular machine. You can see here on the right, it's a large subunit. It has a small subunit. There are the transfer RNAs that we were talking about, and there is some mRNA. So this is actually a crystal structure of the complex where translation is going on. And is extremely difficult to crystallize. So it's a protein with some RNA, and even though it gives rise to all the other proteins, it actually was really hard to crystallize because of its intrinsic structure. It's a complicated mesh. So these are the chicken or egg problems. If you consider the ribosome are made of protein and RNA, how is the first ribosome in a daughter cell generated? And of course, the answer is that the daughter cells have ribosomes that they inherit from their mother cell. But then, how was the first ribosome on Earth made if there was no previous ribosome? Okay, so that question is more at the level of the RNA, and how the RNA actually was one of the first scaffold. But now the real question I want to ask you is, how would you try to find the structure of a ribosome if conventional methods in X-ray crystallography fail? So you know, in that time in 1960s, where there was X-ray crystallography, but it didn't work to crystallize a ribosome. So what would you try to do?
maybe when the ribosome is being made, like assembled, we could look up the smaller parts when it's trying to be, uh, I don't know, assembled together, so that we can study how each one of those components that's getting created folds and turns into this big macromolecule, right?
Yes, yeah, that's a great answer, and that's part of what was done in the end. They had to do it in subunits. They couldn't crystallize everything at the same time. Yeah. Sorry. Any other? I think someone else had raised their hand. Oh,
Miguel,
yeah.
well, I'm actually not sure how she did it, but I believe there's this Nobel Prize chemist, Ada Jonas, that was able to crystallize and get the structure of the ribosome, right? I'm not sure if she was the one that did it in subparts, but I believe I guess that's the reason why she was awarded the Nobel Prize, right?
Yes, that's right. Yes, but so there was someone else, I think. Oh, it's I don't know who it was. Okay. So yeah, well, as Santiago told us, there is the problem for many years, and then Dr. Adayana was inspired in the polar bears actually to solve this problem. So she was working on the ribosome for a long time, and there couldn't be a method to crystallize it. No one could do this, and then she took some break, and then she looked at an article that was talking about the polar bears and their ribosomes. So actually, polar bears, because they hibernate and they live in very cold conditions, their ribosomes are different from other species, and they have a different structure, but slightly different structure and conformation. So based on that serendipity finding about polar bears and the ribosomes, which they didn't have a structure, but they knew that some of the parts were different. She thought of freezing the ribosome to be able to crystallize them. So using a method called cryocrystallography, she invented this new technique in where when you freeze at really low temperatures, you can get a crystal of very complex proteins. And this is how some of the crystals that they got, this as you were mentioning also, someone else was mentioning, there are different parts of the ribosome that had to be crystallized independently. And this was a work of many years. In fact, she would describe this discovery as climbing the Mount Everest, because every time they got to a place where they thought it was the end and there was nothing else, there was another peak to climb and another challenge. And in the end, after several years, there was a structure of high resolution of the ribosome. So this is just a fun thing that, kind of her hair looks a little bit like the ribosome. And well, she even has this picture showing that as well. So I think now is the next. Oh yeah, we're almost in the summary. So just this slide talking that proteins can provide many essential functions in the body, and there are digestive enzymes that help facilitate chemical reactions. There I support the regulation and expression of DNA and RNA. There are antibodies that support immune cells and also transport different nutrients across the cell, hormones, and muscles. So these are really proteins will be as the video was saying, the flesh and blood of the human body. So any recap, we learned how chromatin states prevent all or allow the binding of RNA polymerase and transcription factor. This results in controlled gene expression and is one of the reasons why cells with the same genome can acquire vastly different shapes, functions. RNA is specialized in In addition to messenger RNA, transfer RNA is an adapter for amino acids. The ribosome is the mother of proteins and was crystallized at ultra-low temperatures.